Okay, so I would attribute DNA from the start of it from Frederick Miescher. He discovered the nucleon. He was researching proteins in leukocytes. He discovered a substance that had high phosphorus content and resisted protein digestion. Then there was Aaron Theodore Levine. He wanted to study the chemical nature of the nuclein that Miescher had discovered. And his Levine's first discovery was the phosphate sugar base, the ribose, the deoxyribose, and how RNA and DNA molecules are connected. He also developed the polynucleotide model. We learn that hydrolysis was used to break down and study yeast nucleic acids. That's what he did. He discovered that the nucleic acids were made up of nucleotides, and each nucleotide was made up of nitrogen-containing bases and sugar molecules and a phosphate group. He learned how nucleotides are ordered, and he proposed the tetranucleotide structure, but that was disproved by Erwin Chargaff. He discovered the amount of purines equals pyrimidines. So that's adenine, thymine, and guanine and cytosine. And they pair and they're equal. They pair equally. He developed the research program to study nucleic acids. He wanted to know if different species have different DNA. He learned that through all life, the DNA is different. It's different. Nucleotide composition of DNA is different through all of life, and that nucleotides didn't repeat in the exact same order. He learned that all DNA has the same proteins. It's all made up of the same proteins. And he labels the adenine to thymine and guanine to cytosine as Chargaff's rule. Then there's Watson and Crick, who compiled all the research from everyone, and they developed their own theories and proved them and disproved them. They made the DNA helix structure. They came up with that. They discovered how the chemical components matched up. They learned that hydrogen bonds connect the bases. They learned that there's a 5' and a 3' end, and that the backbones, I think their backbones are parallel. So then it spins or it twists. And then I was reading about Linus Pauling, and now he developed an advanced model building of 3-dimensional structures based on molecular distance and bond angles, and I think he also helped Watson and Crick kind of develop that structure, the model. Then there's Rosalind Franklin. She developed the famous Photo 51. She captured the X-ray image of DNA, which was an X. She did, in fact, help with the development of the DNA molecule, of what we know of it as. although she didn't get any recognition for that. I think it's probably because of that time. Maybe maybe she wasn't friendly. It sounds like Watson had tried to encourage her to work with them, and she didn't want to have anything to do with them. So maybe it's kind of her fault that she wasn't included. But maybe it's, I don't know. She worked at the King's College with Wilkins. She used the X-ray diffraction and studied the structure of molecules. She did write a paper that did play a role in the information that Watson and Crick developed. Also, Wilkins, I believe, gave Watson her photo, and it confirmed it was Crick's idea of what the double helix, that it may be a double helix, and what it could possibly look like. Franklin was really proficient with, I guess the X-rays, but she recognized that DNA takes two forms. So there's the crystalline A form and the paracrystalline B form. She says that the DNA could be in a helix form. Her report states that the A form had symmetry and had an even number of sugar phosphate strands going in opposite directions. Although Crick and Watson say that they didn't use her information, although later then they go back and say that they did. I really like that I had read at one point, which kind of gives her some credit, that Wilkins and Franklin developed the experimental evidence, and Watson and Crick developed theory. I think that kind of puts them as a team there, although luckily now she's getting some of that credit. So that's pretty cool, kind of, women like that, I guess. I don't think she had a very good relationship with her colleagues. Then there is the Griffiths experiment. He used bacteria and mice to develop vaccine against pneumonia. He used two strains, the R strain and the S strain. The R strain didn't cause any illness, and the S strain did actually cause illness. He did inhibit the S strain with heat, and the R strain it didn't cause anything, so he didn't do anything with it. But when the R strain and the S strain were combined, they became active. He studied this, and he concluded the R strain must have a transforming principle from the inhibited S strain. He assumed that bacteria transfers genetic information. Then Avery McCarty and McLeod kind of continued that research, and they wanted to identify Griffith's transforming principle. So they took the inhibited S cells and they destroyed the cellular components, and then they collected the transforming principle. They assumed that could be DNA. They learned that protein and RNA destroying enzymes did not affect the transforming principle, but DNA destroying enzymes eliminated the transforming principle. Avery assumed the possibility of another substance was present, and that was the real transforming principle. So they concluded that DNA is the genetic material responsible for bacterial transformation. Hershey and Chase did experiments based off that. They studied the viruses that attack bacteria. They used protein particles and DNA. Hershey hypothesized that genetic material would be protein. The phages would attach to the surface of the bacteria cell and would inject the cell with DNA or a protein. And then this gave instructions that made the bacteria reproduce. The test included radioactive elements. It was radioactive sulfur and phosphorus. When the radioactive sulfur was used with the proteins, only phage proteins were radioactive. Then radioactive phosphorus was used and produced radioactive phage DNA. Later, the phage parts were removed from the bacteria cell, and this concluded that DNA and not protein were injected into the bacteria cells and made the genetic material of the phage. The DNA was the genetic material. The virus's DNA takes over the bacteria and produces new viruses, supporting that DNA is the carrier of genetic information. Then we have the conservative and the semi-conservative and the dispersive model. The conservative replication is DNA replication, and that consists of both DNA strands and another molecule that consists of two new strands. So one DNA strand makes two copies of DNA. And then there's the semi-conservative replication, which are the two strands of DNA that have unwound from each other and are now templates for synthesis for new strands. So each new double helix has one original strand and one new strand. Then there's the dispersive replication, where the DNA replication results in two DNA molecules that are hybrids of parent and daughter DNA. So that's one DNA strand breaks apart into multiple parts, and new DNA is formed from those separate parts. Then there is the Michelson-Stahl experiment. Michelson and Stahl used E. coli to replicate DNA. The E. coli was grown in a nutrient-rich broth containing a heavy isotope of nitrogen. The bacteria absorbed the nitrogen and synthesized new biological molecules and DNA. Multiple generations of growth passed, so not like years. The bacteria would replicate, and then that was a generation, and then another generation. It was just a replication. That proved that the bacteria's DNA were all heavy isotopes. Then the bacteria were moved to a medium with light nitrogen isotopes and grew for the same generations. The DNA later showed a lighter isotope. Michelson and Stahl purified samples of the DNA and measured density. A single band was developed after no generations passed. This represented the DNA containing only heavy nitrogen isotopes. Only a hybrid band was developed after one generation with medium density between heavy and light isotopes. So that means that these hybrid bands, the model shows two bands, one for heavy molecules and one for light molecules. Two bands were developed after two generations. One was a hybrid band and one was a lighter isotope band. Then after the third and fourth generations, hybrid molecules formed light molecules. Over time, the molecules would become lighter and fainter. This proved that DNA replication is semi-conservative. So the semi-conservative is, remember that each new double helix has one original strand and one new strand. Then there's the structure of DNA. Two polynucleotide chains, including a nucleotide subunit made of 5 carbon sugars. Hydrogen bonds hold the chain together. Sugar is the deoxyribose. Then there's adenine, cytosine, guanine, and thymine. The DNA strand has chemical polarity. So I was reading that it was like holes and knobs. So then they just kind of stuck together. And this is how they lined up. These were the subunits. The three-dimensional structure comes from the chemicals and structural features of the polynucleotide chains. The hydrogen bonds cause the bases to align inside the double helix, and the sugar phosphates are on the outside. The bases pair up, holding the backbone up. The bases are held together by hydrogen bonds, and it's a double-stranded helix. There's a 5' and 3' strand, the 5' end and then 3' end. These are held together by the nucleotides. And then hydrogen bonds on the inside and on the outside allow their chemicals to attach. The 5' and the 3' are carbon numbers in the backbone of DNA. They are developed of a phosphate group. The 5' is a phosphate group, and the 3' is a carbon hydroxyl group. This allows for the direction of the double helix. Then there's the Okazaki fragments. So during DNA replication, there are short DNA segments. There's like a longer strand and a shorter strand. The shorter strand is the lagging strand. The DNA it's in the double helix, so it has to separate. This is the Y, or the fork. That's when the strands are able to, because they're apart, that's when replication happens. The polymerase are the enzymes that develop long chains of nucleoacids so as something comes in, I don't know. The nucleic acid, so when it separates, one side is a template so the DNA replicates into RNA, and then as the codons are read and transferred, a little peptide chain is made. Then there's telomerase. They're proteins that protect the end of a chromosome. They help with cell division. As a cell replicates, the telomeres get shorter until they are so short that the cell can no longer divide. I think I had read that was kind of like a shoelace. It's there, and then that it marks when it's done dividing. That's the end of the codons, I think. Then there's the replication bubble. I think that's what I was talking about with the Okazaki fragments. The DNA separates, but it's not like it just stops. It's a chain, so it has to come out, and so it looks like a bubble. There's two forks, one on one end, one on the other. It's splitting the double helix. So DNA makes copies of itself. The base pairs are connected by hydrogen bonds. Because DNA is copied in the cells, each daughter cell has its complete genome. How genomes control protein function. Proteins identify, maintain, and deconstruct the chromosome. Proteins tell what the chromosome is going to do, I think. Let's see. So genomes are genetic material in the cell. They determine the order of the amino acids that make the proteins. Hmm, amino acids. Um, the structure of DNA solves the stability of life and the mutability of life. So the structure of DNA allows for genetic replication, keeping life going, and there is still room for mutations. Uh, and the mutations allow room for growth and change to take place. They allow for things to be different. Hmm. Gene to protein dogma. The central dogma of molecular biology is the theory stating that genetic information flows only in one direction from DNA to RNA to protein, or from RNA directly to protein. This is not always true because of prions, and they replicate without going through DNA or